This is a production of Cornell University.
Yep, I'm in Rebecca Nelson's lab, and today I'm going to be talking to you all about exploring the genetic basis of Fusarium ear rot resistance and yield component traits. So I just want to direct your attention to this nasty looking ear on the left and these white streaks that are on the kernels. And those are signs of Fusarium ear rot. This is a disease caused by a fungal pathogen, Fusarium verticilioides. It can infect the ear by a few different methods, either by systemic infection or more commonly via the silk channels or injury to the ear, most commonly due to insects. And Fusarium ear rot, we saw those sort of streaked starburst symptoms that are signs of the fungus on the kernels. And it can have a yield penalty just by being an ear rot, but more concerningly is that the fungus also produces these fumonisin mycotoxins. Now, these mycotoxins affect human and livestock health. They're regulated in the United States. This is the range for human consumption. And this fumonisin B1 is the most toxic form. It sort of resembles a lipid and affects lipid synthesis. And so there's, because of this regulation, there can be an economic cost if a grower has corn that's over these limits. Now, these are most commonly found in tropical and subtropical climates, much warmer than we have around here. So most of the work I'll be talking about today, or all of the work actually, was performed in North Carolina. And it is a significant disease in southern United States corn production. However, I think maybe within my lifetime, we'll be doing some Fusarium ear rot trials here in New York as well. Now, this disease has been kind of worked on in our lab for about the past 10 years. And in particular, a past grad student, Laura Morales, was really interested in looking at how this disease related to different ear traits or yield component traits. So to kind of set the scene, Laura had looked at within the Goodman diversity panel and found that in the temperate subpopulation, these yield component traits shown here, clustered here, did not correlate with Fusarium ear rot. However, if you analyze the tropical subpopulation, they're actually positively correlated, indicating that perhaps if you're using tropical germplasm, breeding for increased yield might also increase susceptibility to ear rot, and vice versa. If you breed for resistance, you might affect yield. So that's a little concerning, and Laura looked deeper into this in a 2019 paper where she used NAM families to identify some QTL. And this is a busy table, but I just want to highlight those in blue were QTL that she found affected both Fusarium ear rot resistance as well as ear traits, while those here in orangish are those that were specific to Fusarium ear rot resistance. So she found many QTL, and I've just highlighted a few here that I'll be talking about more later. So this was sort of the platform for me to take off from some of her work and try to really have the goal of better understanding how Fusarium ear rot and these yield component traits are controlled within loci. Is pleiotropy occurring, or is it all linkage of different genes? And also, do we have this sort of only have this Fusarium ear rot ear trait trade-off, or can you have sort of a double win scenario? And then are there different dynamics going on in tropical versus temperate germplasm? So really the basis of my work has been using this nested near isogenic line population. It's developed about like 15 years ago or so by Syngenta, and you can now acquire seed from CIMMYT. And what's nice is that this nil population was developed using B73 as the recurrent parent and 18 of the NAM founders as the donor parents. Our lab acquired these nils in 2 separate sets. And so those 2 sets have been analyzed by Judy Kolkman for northern leaf blight, and she found some nice results for loci that are affecting NLB resistance. And Laura also has a paper looking at the separate set for southern leaf blight resistance. So both foliar diseases, but we've now been able to sort of unify all of these together. And what I'll be talking about today will be the first time we've been using it on an ear rot disease. So to give you a sense of what this sort of nil germplasm looks like across the genome, I'm showing here the introgression tiling path for about 10% of them that are included in the experiment I'm going to be talking about today. Here on the y-axis, each tick mark is just a different nil. Couldn't include the names because there's so many. And then the blue lines are the donor introgressions, which within each of those nils. And so all of the white space is just representing your B73 recurrent parent. And so you can see that even with just 10% of our total nil population, we can get pretty good coverage of the entire genome. Now I wanted to build off of Laura's work. And so for my experiment, I selected nils that were had introgressions within the QTL that she had previously identified that I showed earlier in that table. Here, the bluish highlight represents those trade-off QTL that have affect both FER and different ear traits, whereas these orangish beigeish QTL are those that affect just FER. Now to zoom in on just one of those, we can see that this chromosome 1 QTL here that Laura identified sort of in orange here has 9 nils with introgressions that are contained within it. And what we're trying to see is whether these nils actually contain the variation that she had observed, and can we sort of validate her QTL effect, as well as try to, we have more donors in this population, and try to see if we can understand more about the genetic control of that locus. So our experimental design was to grow all of our nils out here in beautiful Clayton, North Carolina, inoculate them with Fusarium verticillioides. And as I mentioned earlier, there were 196 nils in this experiment, along with their founder parents. And not all of those were included in, were from my experiment per se. Also, Miriam Maun, a former postdoc in our lab, and our lab manager, Ace Repka, also had nils in this experiment. So some of the results I'm showing you are representing nils that they had included in their experiments as well. But they were all grown together under the same conditions and same treatment in 4 plots, inoculated plots, and 3 ears were harvested per plot for phenotyping. So for phenotyping, we just took some visual estimates for disease and insect damage. And we also utilized this flatbed scanner pipeline developed by Nathan Miller at the University of Wisconsin. And so you can see an image from that here. You get nice cob length and cob width measurements, as well as you can get metrics of cob color. And we took some other yield component traits, and we're actually still working through some of this stuff to get their mycotoxin data, as well as to get scanned kernel traits. Now, I've taken a couple different approaches to try and analyze this data. One is a pretty basic one to just look at within a QTL. So for example, within this chromosome 1 QTL I showed you before, seeing whether we can just fit an ANOVA model to see whether there's variation among those nils for that trait. And the other is to take a more genome-wide approach and using all of our phenotyped nils, run association mapping for each of the traits. So slightly discouragingly, this within QTL analysis using ANOVAs to look for variation within each of the traits that Laura had previously found, did not find any significant differences. So that was slightly discouraging. But looking at genome-wide, also for Fusarium irrat, it was slightly frustrating to not find significant results for this. However, when looking at some of our yield component traits, we did find significant hits for Cobb length on our GWAS. Here, a couple on chromosome 1, as well as chromosomes 8 and 10. And I particularly want to note here on chromosome 1, around 50 megabase pairs, we also found a hit for this Cobb red intensity trait. And this Cobb color is a trait that's actually has some major effect loci, unlike sort of Fusarium irrat, which is all quantitative, as well as many of the sort of ear traits, Cobb length, for example. And so here we can see we have some nice peaks that indicate we have some variation in our population controlling sort of some of these contrasts that I'm showing up here in Cobb color. And when you look at some major effect loci, you can see that we can identify some co-localization between many of these cloned major effect genes and our GWAS peaks here. And I particularly want to highlight pericarp color 1, which is previously been shown to be involved in fumonisin resistance. So found it interesting that this region seems to be, although we didn't have variation in Fusarium irrat, we do have a hit for possibly a gene that's involved in fumonisin resistance as well. So to summarize, our scanner phenotyping and that pipeline, along with our near isogenic lines, provide a rich data set to explore these sorts of issues. Sort of our preliminary within QTL analysis hasn't yielded much, but association mapping did find some significant loci that are promising, and I'm interested in following up on, and hopefully more to come as phenotyping and analysis progresses. I want to finish by thanking everyone in the Nelson Lab, especially those bolded who contributed a lot to this work, as well as our collaborators at North Carolina State who helped us with field space and inoculations, funding sources, and of course, thanks to all the beautiful corn that had to suffer for this experiment. So thanks for listening, and I'm happy to take questions.
Sure, that's a clap too. Good job, Will. So it looks like your NIL 1194 has a relatively narrow region when you tile it with the other NILs in your QTL region. Do you have an idea of how many genes are in that narrow region that you could look for candidates?
I don't, and I guess there's also the concern that it might not contain the, I don't know where in that QTL the genes controlling these phenotypes are, so it could be actually not even in that introgression at all. But yeah, not sure how many genes are in there, and not even sure if the genes we're interested are even contained in that introgression.
So to follow up with that, would it be worth, what the NAM parent is for that introgression line. So would it be worth going to the NAM rails and just inoculating them and seeing if you could replicate that QTL region by mapping?
Yeah, so this was, Laura had used the NAM rails to initially identify these QTL. So that's sort of what we're working off of. So I guess that's sort of already been done. Not in all of the families, though, I will say that. So okay. We're including more donor parents in our study.
This has been a production of Cornell University, on the web at cornell.edu.